In the previous lesson, we saw that pyruvate must be converted to a 2-carbon compound before entering the TCA cycle. This is referred to as the transition step, and we will consider it in detail in this lesson. The conversion of pyruvate to acetyl-CoA is accomplished by a large multi-subunit complex known as the pyruvate dehydrogenase complex. The overall reaction is outlined in the figure below. It is an oxidative decarboxylation, with one carbon being released as CO2. There are 3 enzymes involved in this process and 2 involved in its regulation. In the first step of the reaction, pyruvate is decarboxylated by the enzyme pyruvate dehydrogenase, for which the complex is named. This enzyme carries a thiamine pyrophosphate or TPP cofactor. One that we saw in a previous lesson participated in decarboxylation of pyruvate to acetaldehyde. Here again, a decarboxylation of pyruvate requires the same cofactor. The hydroxyethyl group that results from decarboxylation is covalently attached to the TPP cofactor, as shown in the figure below. The purpose of the next step is to oxidize hydroxyethyl to acetyl and transfer to coenzyme A or CoA. This step is catalyzed by dihydrolipoyl transacetylase, which also has a special cofactor. The lipoic acid cofactor is shown in the figure below and is covalently attached to the enzyme by means of the epsilon amino group of lysine. Notice that this cofactor contains an oxidized disulfide bond, and this is essential to its function. This group oxidizes the hydroxyethyl group and in the process becomes covalently attached to the resulting acetyl group. Notice that in the process, lipoic acid is reduced and a thioester bond has formed with the substrate. Notice also that the TPP cofactor has been regenerated to its original form. In the second part of this reaction, as shown below, the acetyl group is transferred to the sulfhydryl group of CoA. Since we broke one thioester bond to form a new one, there is no net energy cost. Hopefully you can see the essential nature of the lipoic acid cofactor. It provides an oxidizing agent that can also form a thioester linkage. Notice that though the conversion of pyruvate to acetyl-CoA is now complete, the lipoic acid cofactor is still in the reduced state. The final enzyme in this complex, dihydrolipoyl dehydrogenase, is therefore needed to regenerate the oxidized form of the lipoamide cofactor. The dehydrogenase carries an FAD cofactor that oxidizes lipoamide and at the same time reduces FAD to FADH2. The cycle is complete when those electrons are transferred from FADH2 to NAD+. The net reaction of this complex is therefore the conversion of pyruvate to acetyl-CoA with the concomitant production of NADH and the loss of carbon dioxide.